Welcome Biology 230 students at Tuskegee University. This is Dr. Griffin with the third video for Chapter 5 of Module 3. Again, Module 3 consists of both Chapters 4 and 5, and Dr. Whittington has completed and posted one video for Chapter 4, and I am doing a set of videos for Chapter 5, and this is the third out of the 5 videos for Chapter 5 associated with Module 3. This video's focus is going to be on proteins. Well, I've seen nutritional facts and you say, oh, your peanuts or your soybeans or your prime rib or barbecue or what have you has X amount of grams of proteins. Well, what in fact do proteins do? Why do we need proteins? Well, proteins essentially provide the main basic diverse functions that sustain life. They are pretty much the action players in your body, in our bodies. They are the main actors and actresses that do a good chunk of all the work, and all the other molecules are simply sometimes modifying or modulating or regulating the activity or the presence of proteins. Proteins are involved as enzymes, so they are going to speed up the rate of reactions in our body. They are involved in defense mechanisms. So think about antibodies and the receptors that those antibodies interact on. Those are proteins. Proteins involved in storage, transport. There's hormonal regulation done by proteins. Again, receptors. These are where molecules actually will bind to the cells on specific receptors that will allow for that message to be received. And we'll talk more about this in Chapter 11 in cellular communication. And there's also motor proteins, so exactly moving and contracting your muscles. Those are all proteins. Hemoglobin, myoglobin, etc. Proteins. So these are what proteins are, which are just the main agents, the main actors for sustaining our lives. Proteins are polymers, so they are true macromolecules, and they are polymers of the monomer amino acids. Here is a diagram of a prototypical amino acid. All amino acids must contain an amino group, which is just nitrogen bonded to 2 hydrogen atoms. They must also contain a carboxyl group. A carboxyl group contains a carbonyl. Listen to me very closely. A carboxyl group contains a carbonyl group. See the carbonyl group? Point to it. Where is it? That's right, it's that C double bonded to O. That is the carbonyl. When we add this hydroxyl group, remember we just talked about carbonyls and hydroxyls in video 2. When we add this hydroxyl group to a carbonyl, we now have a carboxyl group. Okay, I don't take off for spelling unless it spells something different. So if you put carboxyl and you really mean carbonyl, I don't know to make the differentiation. So it would be wrong if it was the incorrect answer. The main group is this side chain of amino acids, and this is what's going to be unique to all of the 20 amino acids in our bodies. This is what gives the amino acids its character, its flavor, if you will. There are 20 main amino acids in our bodies that have 20 different R groups. These R groups can be polar, they can be nonpolar, hydrophobic, hydrophilic, etc. So whatever the R group is for your amino acid, that's going to be the flavor, the quality, the makeup of that amino acid. Okay? The R group gives the character of the amino acid. I'm not going to put all the 20 amino acids on here, but you see it in figure 5.16, figure 5.16, and it details and groups all the 20 amino acids. So you should be able to look at an R group and tell me if it's polar or nonpolar, or if it will be hydrophilic or hydrophobic, etc. The amino acids are joined together when we're making peptides, which eventually will make proteins. They're joined together by peptide bonds. So remember, let's say we're going from monomer to polymer here. We're forming a new bond. We're removing this water, this hydroxyl group and this hydrogen group. They're going to be removed through, what's that reaction called? That's right, we're removing water, so it's going to be dehydration. Okay, remember that's from video 1 for this chapter. We're removing water through dehydration, and we're going to join this carbonyl group with this amine group here, forming this peptide bond. This peptide bond is where the carbonyl group, get my arrow back, where the carbonyl group joins this amine group here. And this is the peptide bond. So you should be able to recognize and visualize peptide bonds. There are 4 levels of analysis in which we examine protein structure. And these are highly detailed, and these figures in your book are excellent. 5.20 and 5.21, these are excellent figures. Go over them in very good detail. Make sure you understand each level of analysis. Okay, so the first level of analysis for protein structure is primary structure. This is the simple amino acid sequence. Okay, these are just the letters that represent each amino acid. So if I ask you for the amino acid sequence and I give you a whole set of criteria for a protein, I don't want to see shapes. I don't want to see sugars or features or structures. I want to see words or 3-letter abbreviation. Example, glycine, leucine, isoleucine, alanine, etc. The primary structure is the sequence of amino acids. The secondary structure pertains to the folding and coiling patterns that are going to stabilize the peptide backbone. So how are these amino acid sequences structured on a very basic level? And the main types that we'll discuss and that your book discusses are the alpha helix. And that's just the coil. You can see this in figure 5.2 and 5.21, as well as the beta sheets or the beta pleated sheets. So those are just 2 different types of secondary structures proteins can have. At the next level is tertiary structure. Tertiary structure is the overall 3D shape of a peptide. Okay, and a peptide is a string of amino acids. Okay, examples of tertiary structure is disulfide bridges, so how this peptide is going to fold on itself, as well as hydrophobic interactions, which also gives you an idea of how a peptide is going to fold within itself and on itself. Last but not least is quaternary structure. Quaternary structure describes the overall protein structure. Now the main difference between quaternary and tertiary structure is that quaternary structure describes the interactions between multiple peptides. I can't say that enough. Quaternary structure, interactions between multiple, meaning more than one, peptide. So if you have a monopeptide and you only have one peptide, you will not have any quaternary structure. However, most proteins have multiple peptides within them. Different subunits representing different sets of peptides. Most of the channels in your nervous system are composed of multiple subunits, multiple peptides, and how those different subunits interact with each other and are structured will give the quaternary structure. Please make sure you have that clear in your head, okay? This is the end of video 3 out of 5. Okay, you are 66.66666 to infinity % done. Okay, you only have 2 more videos to go. I know you can do it. This is Dr. Griffin signing off. I look forward to looking and learning with you on campus and in class and presenting the next 2 videos for Module 3, Chapter 5. Have a great day.